Hello, everyone. Welcome to the Sensory Systems oral and featured oral presentations of Computational Neuroscience 2021. I'm Julie Haas. I'm a member of the program committee, and I'm here to moderate this session. Our first talk this morning is from Evie Malaya, and she will tell us about processing sign language. She is our featured oral today. And without further ado, here is Evie.
Thank you very much. Let me share my screen and let me make sure that I'm able to share the actual presentation.
Okay. Let me try to focus you. This isn't working. There we go. Great.
Okay. Do we have it?
Yes.
All right.
All right, good to go.
Okay. Today I will be talking about something slightly peculiar. I will be talking about language processing, but in a completely different domain than we're used to. I'll be talking about processing of sign language. And what we found when we started trying to figure out how the processing of sign language works in native sinus. So this is a collaboration between multiple universities in the States, in Salzburg, and I'm actually at the moment in Germany at the University of Freiburg, where I'm working with a team that looks at entropy processing and language. So let me try to make sure that I can switch the slides here. There we go. Okay, so being trained as a neurolinguist, I have very standard picture about how language processing usually is expected to proceed. We first have an interpretable signal, something that the speaker or the signer can understand in the native language. Then the speaker or the signer will zero in on information-dense regions, and we'll talk a little bit about what that looks like in auditory and visual domain, after which perceptual parsing and matching of lexical templates, your lexicon matching with what you hear or see happens. And finally, comprehension really should consist of neural modeling of what it is you see, and that's when you actually comprehend what's going on. Just to give you an idea, this is really not what we found is actually happening. So let's see what we actually found. So a quick reminder, what we're looking at when we talk about information-dense regions, we're talking about Shannon information. So the more potentially variable next item in what you see or hear is, the more potential information there is in the message. So if we look at zeros, ones, there's really 50-50 probability of what you see next. When we add the digits, we get to 10% probability of what you see next. With hexadecimal system, it's even higher, and so it goes on. Now, what does Shannon information look like in auditory modality as time series and in the visual modality? So this is actually the best presentation I've ever found of looking at what high complexity looks like in auditory domain, so acoustically, musically. So that's our leftmost column. And the high complexity static visual signal. If we try to plot both on a 1/b to the beta scale, what we end up with is completely, scattered plot. If we look at low complexity, this is the rightmost column, both visually or acoustically, we get something that's really boring, looks very linear. Humans in general tend to prefer aesthetically, and it seems in language, something in between. So moderate complexity seems to be our preferred mode of processing. This is where people's aesthetic peaks tend to be. This is where language tends to sit. If we try to plot the language on a 1/f to the beta is somewhere between 1 and 2. Now, trying to move to the dynamic visual complexity, which is a little funky to think about, what we did to understand it, to quantify it, was to start looking at the optical flow. So we started with the videos, actually. So you can think of, all right, video is two-dimensional plot of a three-dimensional action. And if we have time, I might be able to show you an equivalent of optical flow in radar data, which is three-dimensional. But looking at optical flow basically allows you to look at how far each individual pixel has moved between two frames in the video. So the frequency of optical flow obviously depends on what video you're looking at. Here we have a person putting together Legos. This is from Jeff Zach's lab studies. And then we have a signer giving instructions in American Sign Language. So if we plot these and in the time domain, what we see is, here on the x-axis is our time in seconds. And on the y-axis is bin optical flow. So the lowest bin is the fewest amount or distance of pixels, how far the pixel has traveled, and the higher you go, the longer the bins are, so the further distances. So if you just look at the two pictures, what you see is, first of all, everyday action seems to be kind of bunched into put together things. So if you try to ask yourself, well, what is the variability? What is the entropy in the everyday action versus American Sign Language? You can say there's more going on, dropping off here and there in American Sign Language. So if we try to actually plot that spectral density across multiple videos, multiple things that people are doing, and multiple pieces of sign language conversation, what we end up with, averaging everything out, human motion, is less complex than signing. So beta in signing ends up being higher, and sign language is more complex. This is already published data, but what that told us is, if we're looking, given the frequencies that we're looking at, so this is NTSC system, so 30 frames per second, we have to go down to 15 at our Nyquist frequency when we look in the frequency domain. So in general, the information in sign language is sitting across all the frequencies. And there's more of it. So even if we just look at the optical flow, which at this point we're thinking, two-dimensional reduction in a sense of the sign language signal. So that's pretty interesting. So what we want to do after that is figure out, okay, people are perceiving that information. Well, how are they parsing it? From non-signing work, we already know that humans who do not sign tend to parse their reality based on the velocity, acceleration, and direction of movement around them. We are, like all animals, very, very sensitive to movement. Those of you who have dogs probably know that dogs will notice very easily a moving animal, but if there's a deer just standing in front of you, the dog might not even see that deer. So we're very similar like that. So here, again, Jeff Sachs' work, who works on perception, humans are trying to parse out visual scenes into segments of different actions. And people agree very clearly on what they see, even if these things are just dot point plots. And humans tend to rely on velocity, acceleration, direction in that kind of parsing. A very similar thing happens when we try to parse out speech. In general, this is work of Keith Klunder from Purdue and his team. They were looking at, in general, what do we need to know from the auditory signal in order to parse it out? And what it seems like, we don't really need to know specifically what vowels there are in the syllable, what consonants there are. What we need is to have that energy peak in the syllable. What's remarkable is if you actually flip those peaks around, so long as the peak is still there, the speech still sounds comprehensible. Again, this is not my work, but this is showing that humans do tend to rely on spectral entropy fluctuations as they're trying to decipher the signal. So what do we get in terms of spectral entropy fluctuations in sign language? This is an attempt of us to try to figure out that kind of fluctuation. Now, in general, this is a little bit difficult because signing tends to have one syllable within each sign, and there's a lot of direction changes going on within the syllable. So plotting that within 2D doesn't necessarily work. So at the very bottom line on the x-axis, the greens are starts of signs and the red dots are ends of signs. And as you see, when we do parsing in 2D, we seem to parse out several syllables per sign because of the directionality changes. Well, let's try to see what happens when we go to the neural modeling. When Dr. Zinker said on Saturday, well, let's, I really hope somebody does this in language because it's easy to manipulate language. I was a little bit shocked because I said, yes, we did that. So what did we do? We grabbed the videos of sign language sentences and we flipped them. So this is one sign language sentence. This is Austrian Sign Language. You know, two girls and one helped the other. And this is the same video, time reversed. So in some sense, the lexical items are still there, but the motion is completely different directionally. And as you would expect, it makes very, very little sense to signers. So presenting multiple, so we had 40 stimuli of different sentences, and we had 40 time reversed videos. Looking at the optical flow of those are very similar to American Sign Language. And when we plotted the power spectral density of optical flow of direct and reverse, as expected, they pretty much overlap entirely. We also asked the signers to tell us whether these are well-formed sign languages. So these are our behavioral responses. On the time scale, people react about the same time, take about the same time to respond. With regard to acceptability, sign language is very well understood. People can say what's going on, who did what to whom. With regard to reversed videos, it's not like they're not trying. In general, they're saying, no, this is horrible, horrible to see, but they're identifying here and there lexical items. So this is some of the acceptability that shows up as high. But in general, there's comprehensible sign language where you can tell syntactically this is a sentence, and then the reversed ones are not comprehensible sign language. EEG data looks very generic. So this is 26-channel EEG with active electrodes, 500 Hertz sampling rate, which we had to filter because, as you'll see later, we needed to scale it down to our optical flow frequency. And then we also did bi-region breakdown, checking out anterior, posterior, left and right segments with 5 electrodes, because what we wanted to know was, well, which portion of brain region, in general, talking about scalp localization rather than source localization, which portion is relying more on which frequencies, if there's any differences as to what's going on. So the analysis then proceeded along the two sides. First, we did the coherence calculation between the stimuli, the optical flow time series, and the EEG. So grabbing the data from 0.2 Hertz to 12.5, because that's our PALAS system, 25 frames per second video frequency. We did canonical correlation analysis in MATLAB, and that's also already published work, by item approach. So we weren't averaging things around. All we did was for each stimulus video, for each electrode, and for each participant subject, we looked at what the maximum peaks in coherence were. Now, once we got those peaks, we also looked at where they tended to occur. In general, they tended to occur between 100 and 200 milliseconds post-stimulus. That is fairly typical for information extraction from video stimuli, and that's work of Green and Hansen, I think, last year. So that told us we were in the correct ballpark in general with regard to calculating our coherence. After that, what we did was we actually wanted to do, I think it's called intelligent AI, but basically we wanted to do classification of the data to figure out whether from this coherence data, if people were looking at sign language or reverse sign language, non-comprehensible data, and then try to figure out, well, based on what could you do this really well? Which features, which frequency bins of coherence could contribute to highly accurate classification? And just as we got those amplitude peaks in coherence along frequency bins, the first thing we did was to actually look at the data. This is the cross-correlation matrix for those peaks in frequency bins. The bins are 0.2 Hertz. Obviously, the diagonal along the center, that's a perfect self-correlation. These are the same parameters that are being entered. What kind of draws one's attention is the fact that early on, up to about 4 Hertz, what we see is we have a lot of positive and negative correlation. So there seems to be a lot of information sitting there in general, whereas it seems to be a little bit more washed out later on. Okay, so we first did a very standard machine learning approach analysis for the whole brain and by brain region. Now, as you see, the accuracy in percent, and this is the accuracy on the holdout set. So the standard pipeline proceeds with taking 20% of the data, putting it aside as the holdout set, and then doing 10-fold training on the rest of the data. The standard algorithms, so this is linear regression, linear discriminant analysis, k-nearest neighbors, trees, Bayes, and support vector machines. They are all sitting in the upper 90%, most with 100. This is a lot. This is not typical at all for machine learning performance on data, much less very noisy brain data. And of course, yes, I understand we're feeding in both the neural response and what that neural response is too, and that the accuracy is crazy high. That tells us, ooh, maybe there's a lot of information in our data. Can we degrade the data and still get pretty high accuracy? This is what we did. So we, instead of putting all the parameters in, we broke down the data into pieces along frequency bins. And as you see, obviously earlier frequency bins are giving us much higher accuracy. So this is on the whole brain. We actually did it broken down by region, and the difference really wasn't that great. So in general, it seems that yes, the information about what's going on is sitting in the lower frequencies in general. So that tells us that there might still be redundancy in the data. And what we want to do is we really still want to figure out, well, where is the most informative part of what's going on? So that's what we did by playing around a little bit with the linear discriminant analysis, turning it into sparse LDA. So basically, as you noticed, we had a ton of predictors, so all our frequency bins were predictors, and number of samples, so 24 participants and understood and misunderstood information, that was not a lot of instances. So by breaking down this data, we wanted to see what could be gotten rid of. So there were a couple of approaches. One was introducing elastic net penalty, and one was introducing group lasso penalty that came in later. And this is both published this year recently. And then this is Katie Ford's doctoral dissertation this year from UA. But fundamentally, after we introduced that penalty, that allowed us to zero out a lot of the parameters and trained on a random sample of 12 observations. So this is just a quarter of the data. Testing on the remaining observation repeated 10 times. The average accuracy that you see is the average accuracy over those 10 tests. We still get insanely high average accuracy. Well, based on what do we get it? We know we zeroed out a ton of the parameters. And what helps is the entry-wise product. So this is, we're multiplying the coherence value with the discriminant vector for that value. So what is giving us this insanely high accuracy? So you see the CDLR with elastic net penalty on the left, and across front, posterior, left, and right regions, you see that lower frequencies, mostly up to 2 Hertz, but 4 Hertz and maybe a little bit higher here and there, are still giving us the most information. Note that it's very similar across wherever we're looking. And the function of brain regions when you process sign language is very different. You would be extracting features posteriorly, you would be calculating syntax on the left side, you would be processing the spatial dimension on the right side, and you would be doing executive control in the front. What's going on? So assuming we're dealing with group sparse information, we go to the group lasso approach. And group lasso zeroes out everything that's outside the main group. Using group lasso approach, what we get is huge usefulness of posterior vectors in the first 2 Hertz. And then a little bit on the left, which is obviously needed there for syntactic processing, but it's still extremely low frequency. And we've actually broken down things by region as well, just to be sure that we weren't completely crazy. And we're getting the same result. What does it all mean? So mathematically for you guys, this is probably, sure, this is what you would get when you see a lot of prediction. But when I'm thinking about this as what's going on in comprehension, as a linguist, that tells me that comprehension is not based on sequential processing as we are usually thinking about it in linguistics. Instead, what's going on is prediction. So what this means is comprehension is matching of observed signal to the predicted signal, not to something in the memory, not extraction of templates from the memory or possible syntactic structures. It's you can comprehend the information only as long as you can actually predict it. Now, if we step back and think about it in terms of how does language knowledge proceed, right? If any one of you has tried to learn a foreign language as an adult and pass the exams, you know that the step A in the European classification takes forever. Yes, you're learning a lot of new words. Yes, you're presumably learning a somewhat different syntax. But then if what you need to do is to learn to predict the incoming information based on information that's already there, that's a crazy amount of statistical learning. This is not the same as storing a template for a word in your memory. This is learning to predict that word as it's coming next. So intuitively, this is not crazy. From the standpoint of the basics of neurolinguistics, I'm afraid we are really shaking things up a lot. And right now we're trying to look at what's going on in bimodal bilinguals, so speakers and signers. And those of us who have no idea what sign language is, how do we parse the signal? What would that tell us about our general visual ability? So stay tuned for the next exciting episode. I'm going to try to stop sharing if I can switch the screen up. Oh, great. And this is thank you very much to everybody who's worked on it. Julia Krebs in Salzburg is the sign language specialist. Sean is interested in explainable AI. Katie is the mathematician who came up with this sparse analysis, with Brendan Ames being her dissertation advisor, who was very wide open to applying this to anything under the sun, including neuroscience. Okay, so there we go. I think I stopped sharing, right?
Yes, we're good. Thank you very much for your talk, and thank you for keeping us right on time. So I think, so we haven't had any questions posted yet, so if everyone wants to post questions, that would be awesome. I'll ask a question in the meantime. It's kind of a naive question, I guess, but so your frequency breakdown suggests that we should all slow down?
In what way are you thinking? So yes, one person who should slow down first is probably me. Yes, I do have a tendency to speak very fast. Well, in general, actually, people do have preferred rates of speaking. Normally, what we see is about 4 syllables per second. So you'd have about 250 milliseconds per syllable, and that's the average preferred rate of information transfer.
And you're talking verbal listening speech or sign language?
The duration is actually about the same. So you get about 4 syllables per second. Younger folk tend to go faster. As we get older, we tend to go slower, and we also do prefer people to speak to us a little bit slower. You probably noticed the younger generation also speeds up any of the YouTube videos. That's pretty fascinating to me, because I find I can't process things that are at 1.5 speed in general. So I don't think that necessarily means that we need to slow down, but what that does mean is that the amount of statistical learning that is necessary for correct prediction takes forever. And you've probably had a sense of, so English is not my native language. And as I speak, people normally have kind of a bit of a feedback going on in their head, as in, I predicted something similar to what she would say, but her vowels are all over the place. And so it's a little bit difficult to keep up with everything, because the predictions consistently fall a little bit off from where they should be. And the comprehension as it should be happening, so we're used to understanding things as they're happening, right? Well, that's our predictions being correct. When our predictions are incorrect, the catch-up can happen, but then we lose out on the incoming signal. And so I think we're not realizing how much we're asking of our brain, if that's what's actually going on. And honestly, I am fascinated with regard to, well, how does the learning of a language take place given how much you need? And I understand that consistent statistical input is going to have learning occur beyond one's intentional desire to learn a language. But that's still completely fascinating.
It is, and maybe that explains why when we learn languages, we have to do Affirmative Book first and very slowly. Again, slowly. We have a question from Milena, who I will invite on screen. And that always takes a second to invite someone on screen. I'm not sure that inviting screen on is working. Oh, Milena's, okay, she's commented. Sorry, can't use her camera. So I'll post a question. So how generalizable do you think the results are for sign languages that are based on different proto languages? Maybe you could explain what those are, which I imagine might have different structures.
Milena, can you type in what you're referring to with regard to proto languages? Because sign languages, so there are sign languages at different stages of development. So Austrian Sign Language is already really well developed. There are sign languages that are kind of in the process of developing. So Arab Bedouin Sign Language, for example. And so I'm not entirely sure what you mean in terms of what might be helping language to develop, the relationship between iconicity of the message and the linguistic structure, because that's kind of a separate book, honestly, all on its own. It has less to do with the frequencies at which we're looking and more to do with the conventions, I guess, in general. So I'm not entirely sure if that's what the question is about. Also, with regard to languages that are sometimes developed in the process of home signing, for example. So parents don't know sign language, the child is deaf, the best input is sign language. So parents start to either learn sign language, which they're not very proficient with, or they try to develop something on their own, such as Nicaraguan Sign Language for a long time was really home signing in individual households. So they start much slower, yes, of course, because they are really more kind of systems of information transfer. But then as kids pick up on those systems, the same process happens as we see with pidgins and creoles. You might be getting as input a system that's not fully fledged language, it's somewhat degraded, and then the brain is going to develop a linguistic system out of it, and it speeds up in the process. But that's kind of more of the evolution of language, less than about processing. I hope I answered the question.
I hope so. I'll ask something else that you just reminded me of. So twins often develop their own language in early childhood. Have you studied those? I mean, that's kind of an open question, but
they often do. I incidentally have twins. They did not develop their own language. I would have studied it with a lot of interest and joy, but no. So they can, yes. And that again goes from the system of signs that can very easily acquire linguistic properties if they're used a lot. So if twins are left on their own with each other, they have to communicate and they will develop a language very quickly. If they are bombarded with other languages all around them, it's much, much less likely to happen because they're dealing with that information dense input signal that they have to handle, and they simply don't have time to deal with developing their own language.
I always found that an interesting aspect. All right, thank you so much for all of your interesting work.
Thank you very much. I appreciate you moderating it.
Yeah.
Thank you. I'm going to close the video.
Okay, great. So at this point, I will invite our next speaker. On screen, we're a little bit ahead of schedule, and that's, I think, okay. Oh, there's another question for Evie, actually. So, and now I've had Mario on.
Hi. I'll close the video. So for the last question.
Yeah, actually, we're a little ahead of time. So just so in case people are tuning in to see you at 10, maybe we'll invite Evie back on screen to answer. Answer the question. Evie, hello.
Hello, sorry,
Okay.
I'm back. I'm just trying to read the question also.
Yeah, so I'll read it out to give you a little bit of thinking time. Does this mean, according to the hypothesis, that if we are learning a new language, watching and listening to a native speaker, even if the language is above our level, will implicitly increase our learning ability on the long range due to improving our ability to predict the stimulus syllabus?
Yeah, so a couple of things here. So yes, anything from a native speaker is best. Something from multiple native speakers is even better, because what you're trying to predict is the general statistical parameters of the signal, right? If you have it with the same person all the time, with the same teacher, then what's going to happen is your statistical parameters of prediction are going to be skewed toward that person's way of talking. And I don't know if you've had an experience. So my American Sign Language ability, I was taught by a female. And I'm actually told that my sign language, which is not native at all, apparently my American Sign Language has a female accent. Whereas some of my colleagues who learn from males, even though they're female, would have a male accent. So,
tell me about that. What is a female and a male accent comprised of?
Well, we don't know. We're studying that using motion capture at the moment. So there's something about the style of motion in sign language. The signs are exactly the same. The syntactic structure is exactly the same. It's something about the way in which you do the either hand internal motion or gross motion that is either typical of males or typical of females. And for signers, it's very easy to tell who your teacher was, as in gender. Because as you're learning, you're really not trying, if you're not exposed, and I wasn't, to multiple signers, you will pick up these statistical regularities that are particular to the language, but also the regularities that are particular to the person.
Okay. So sorry, did I derail you from answering the question about learning ability?
Sorry, I'm trying to figure out what else is going on here. Sorry.
It's if you click on answered, it's the question is there.
Oh, I see questions showing up in the stream too. What if you learn from a male teacher who had a female accent? You'd probably learn the female accent. So it's really the input that matters, not what style of input you have.
Yeah, I think Abdullah was asking whether that implicitly increases our learning ability on the long range.
In terms of, so what do you mean? So having multiple speakers of a language around you, it increases long-term ability to learn something? Absolutely, yes. The more signers of a sign language, the more speakers of a spoken language you have around you, the more statistical information your brain collects besides what you're trying to do yourself. The easier it is to actually get to correct statistical prediction and therefore, as it seems, comprehension. I don't know how much. I think I'm the most shocked person at my own findings than anybody else, because really this is not the way linguists think about it. But both from Dr. Zinger's talk on Saturday, from yesterday's keynote, it does seem that predicting information is what our brain wants to do, even in everyday communication.
Fascinating. Well, thank you again so much for your time. This is such a novel topic. I really appreciated it.
Thank you.
All right, so with that, we'll re-invite Mario back to screen.
Hi.
Hi, Mario. All right, thank you so much for your flexibility. So this is Mario Panunzi, everyone. He's going to tell us about non-synaptic interactions between olfactory receptor neurons and how that alters processing. Mario, if you want to take it away from there.
Thanks. Good morning, everybody. And I will share the screen. Okay. I suppose that you can see now?
Yes, great.
Okay. Okay. Yes. So thanks for coming to my talk. And now let's talk about something quite different. So quite a change of topic. But before starting, I would like to thank my funding that is coming from the Leverhulme Trust, and of course my university that I'm here for the last 4 years. But most of all, I would like to thank my professor and supervisor that trust on me and trust on these ideas that I will present now. So what I will talk today is about something very specific but general for the insects. And but before starting, let's talk about what is quite in common between these two images. That is just some cables covered by rubber and the myelination in a cable of neurons. So we have here both protection from anything that can happen, and we are isolating the information in a way that it is not disrupted by the other signaling happening in the close by. But what happens for this neuron is something slightly different. So we are talking about insects, here the Drosophila in particular, and in the antenna of the Drosophila there are many hairs, and on these hairs there are between 2 and 4 olfactory receptory neurons. And of course, on this small location, there is no isolation, and therefore there is an inhibitory interaction. So for example, in this picture, we can see a resuming of the very nice result from the group of Striatal, where they presented the heptanon that was very that the AB3A was very sensitive to, and on we can see that is inhibiting the AB3B, or the other round, sorry, but you get it. So one is really excited and the other is inhibited. They demonstrated that this effect was really specific on the incoming of the odorant, and moreover, on a later paper, they show that this effect is an electric effect by setting a tungsten cable through 2 sensilla and showing that the same effect can happen. So now the question is, why is it happening? I mean, we have typically myelinated neuron and axons. So why it is happening now? So before to start to answering this question, we have to take a step back and think about the function of the olfaction. So for insects, but in general, it is useful to identify odors, identify odor intensity, of course, and even the ratio of the intensity of the odorants. So we can have in the, for example, in rose, more than 400 different volatiles and different odorants. And usually there are few of them that are really important for insects, but of course we can imagine that is not the intensity of some element, but is the ratio between the concentration that is coming that is important. And then of course identifying the source location and other functions. So this is important to have an idea of what we are looking for. And therefore, it gives us the possibility to have the intuition of 3 hypotheses that has been put up to explain this strange behavior in the Drosophila. That is, the first hypothesis that this non-synaptic interaction between neurons can help to improve the ratio concentration or the correlation between 2 odorant concentration, or to increase the dynamic range of the neurons. These are the idea, but let's see in detail what we mean. So for the first hypothesis, let's think about 2 olfactory receptor neurons and 2 odorants, just for simplification. And let's analyze the hypothesis that we fix the value of one of the concentration of one of the 2 odorant and explore different concentration for the other. We can imagine that in this situation, for the typical response of the neuron, we have a plateau, of course, for higher concentration, and therefore we don't have a perfect frequency rate encoding for the concentration ratio. Therefore, a small inhibition on the other neuron can improve and straighten this encoding and improving it. This is the idea. We will see later a little bit more. For the second hypothesis, we have to talk about 2 important steps. That is, first, when we analyze the 2 odorants coming from the same source, they will have, as has been demonstrated empirically, a higher correlation than odorants coming from separated sources. This is quite intuitive. And the second aspect is that for many insects, this element is really important, and they are behaviorally prone to respond to this element. For example, Baker et al. More than 20 years ago showed that for moth, even a single millimeter of distance between the sources can be perceived even at 1 meter distance. This means that it can be perceived and it is important. Therefore, we can imagine this kind of encoding. So we have 2 odorants coming from the same location. They will be highly correlated. Therefore, there will be a high interaction and less activity. On the contrary, when we have separated sources, less correlation, and therefore less non-synaptic interaction and higher activity. Of course, this is just a cartoon, but we will understand a little bit more later. And for the third hypothesis, we just need a single odorant for the 2 oral ends, and there think about the receptive tuning curve of the olfactory receptor neuron, again on rays, and the plateau that usually happens for higher concentration. The idea is that the non-synaptic interaction can decrease this curve and diminishing the ceiling effect, therefore increasing the dynamic range. Okay, these are the 3 ideas. Let's see how to deal with them. Let's go back to our antenna and see some of the details that we want to analyze. So we know that on these antenna, the olfactory receptor neurons are stereotypically co-housed on these hairs, and they are always connected to the same receptor type and always connected to the same glomeruli in the antennal lobe. To have an understanding of the typical olfactory system, in insects, it is for the first layer of neurons is really after the receptor transduction, and after that we have the antennal lobe, and after that we will start to have the mushroom body or the local horn that are more higher areas, but we will not deal with them. This is the corresponding of olfactory bulb, the antennal lobe. So we have this idea, and what we simulate is something a little bit more simpler. And we just deal with a single air single sensillum with 2 receptor types, and of course 2 types of projectory neurons and lateral neurons. The projectory neurons are the neurons that are excitatory and will give the information further, while the lateral neurons are inhibiting the projectory neuron. This is the schema that we will use, and for now, without entering into details, we used leaky integrated fire to simulate the neurons. And before going further, we said that we constrained the model with 3 important information from the literature. First of all, that with a given input of 500 milliseconds, has been shown that the shape of the olfactory receptor neurons is intensity invariant, meaning that if we normalize for the peak, they have always the same shape. And then we reproduce it. And we reproduce even the qualitative shape with respect with 3 different input from step, ramp, and parabola from the work of Kim and Lazar. And finally, from the work of Olsen and Wilson, we reproduce the function between olfactory receptor neurons and projectory neurons. This is our constraint for the model. And now let's start to analyze the system. So we are interested on the functionality of the NSI, and therefore we will compare to a control model where there is no NSI but no inhibition even at the level of the antennal lobe, and even with the case of the inhibition through the antennal lobe, essentially to demonstrate that not only the NSI can be important, but maybe more important than lateral neurons, especially because in literature has been shown several times that they can be useful for these 3 hypotheses. So with these 3 models in mind, and we stimulated with just the pulses in this case of different duration and concentration values. And as we can see from this picture, for an increasing level of concentration and different value of concentration ratio, we can see, for example, on the control model, that neither the olfactory receptor neuron nor projectory neurons can encode properly the concentration ratio. The lateral inhibition in the antennal lobe improved a bit on the level of of course, the projectory neurons, but we can see that NSI model can improve much more. This is the first step, and I think that it kind of indicates the idea that, yes, they can be helpful in this direction. Let's go on the second hypothesis. We used here realistic plumes. So we simulated these plumes with some simulating the typical statistics shown in literature and observed data. And again, we use the 3 different models, so the control, the NSI, and the lateral inhibition. And we can see here that increasing, so what we want to demonstrate is that it can be encoded the correlation. So for increasing value of the correlation, what we wanted to see is that the projectory neuron activity can encode this information. So we have that the control, of course, there is no information at all, so the level of projectory neurons is always the same. But both for lateral inhibition and NSI inhibition, we can see that it is encoded, this information. We have analyzed more in detail this feature and show that the NSI is slightly better in the lateral inhibition using these 2 features, that is the distance at 0 correlation and its distance to the control, because of course at 0 correlation we don't want any disruption of this effect, and we want that there is a big distances between 0 correlation and correlation high. This is for the second hypothesis, and now let's go further to the third. We have here a single odorant and 2 different receptor neurons. Of course, we removed the antennal lobe as we don't need it now. And for this case, we analyzed the dynamic range as the distance between the first point when we have above 10% of the maximum of the 2 until the point to 90% of the maximum of the 2 neurons. And we can see here that the control model is slightly better than the NSI model. And we analyze the system for different values of the similarity between the 2 oral ends. In this case, we even see that NSI, for very similar receptivity of the neurons, it is even worse than the single neuron. So to recap our analysis, we have seen that the NSI can be helpful to encode the ratio of the concentration, the correlation of the concentration, and but it is not good to increase the dynamic range of the neurons. After that, we thought that these are very nice hypotheses, at least the first two, and in these years we are trying to test it, and so we are now leading a project where we will do both electrophysiology and behavior and the model. And in the electrophysiology and the behavior, we will analyze the behavior of two genetic modification of the flies. One of the Reaper, and the other is the TNT. And the idea is that to analyze for different level of separation between odorant sources, and see whether the Reaper, that is ablating completely one of the two neurons, or the TNT, that is interrupting the information from the neuron to the antennal lobe, can be different. In this way, we hope to demonstrate that not only that on the model, but even on the reality, let's say, we will be able to that this function, this NSI, can be helpful for the flies. And we will do it again with Thomas Nowotny and the group of George Jimenez, Lydia Ellison, and Andre Maria Chargas, and Claudio Alonso, and from very far, Paul Shiska. And I would like to thank all of them that I'm collaborating with. And that's it for myself. Thanks.
Thank you so much, Mario. Great talk.
Thanks.
So while we're waiting for other questions to appear, I feel like I missed a little bit of how your NSI is implemented in your model and what biophysics you base it on.
Yes. I completely removed from any mention on the. I removed. So what we used is a leak integrate and fire for the neurons. Mm-hmm. And we implemented the interaction at that level, so through the reversal threshold. So the input is coming through a transduction function, and through the transduction function, we manipulate the reversal potential. And this is not at all a mechanistic explanation. We just reproduced the data from Striatal, from that paper, and we just came up with this hypothesis. So completely ad hoc for the moment.
Okay, so it's like a leak channel.
Yeah, and for now there is no information at all of how this mechanism can happen. Actually, there is still debate about how much can we? It's still quite unknown.
Can you talk a little bit about more why you expect the TNT or Reaper mutants to do this differently?
Yes. Can I come back to the?
Sure. I think you can.
Probably can be useful. Okay.
There we go.
So yes. So what is the element here? We know that there is this interaction at the level of neuron, but of course they are this information is going through the antennal lobe and then going on. So just removing and ablating one of the two neurons, of course, remove completely the activity and the NSI. But we want to compare with something. We want a good control that is just removing the information going to the antennal lobe, even though we still want the interaction. So I will try again. I can see that you're still. So for the TNT, we still can see the interaction between the two neurons, while for the Reaper, we will remove completely the interaction. Therefore, this one it will be the perfect control. So the comparison between these two models.
Okay.
Okay, that's
Sounds
the idea. And of course, this is doable practically only with the Drosophila. That's why it is a very good animal model.
Yeah, that's a pretty nice setup.
Yeah.
Do we have any other questions from the audience? We have one. So Martin, who I think doesn't want to come on screen, says only some pairs of odorant receptors are coupled this way, right? Is there something special ethologically about these pairs, or the odors they respond to? Do the corresponding odorants tend to occur together? Do they tend to be correlated?
Okay. So in the world of the, I'll try to answer. They are both questions. So in the world of the insects, they are all paired. And actually, as much as we change the insects, we can see even pairing of multiple receptor and therefore neurons. So, for example, bees, I think that they have even 14 or more neurons or on the same syncytium. This means that there is a lot of interaction there. And actually, that's a very good following up for our model, because, of course, this is a very toy model compared to that one. And for the Drosophila, practically all of them, they are paired. So, and at least 2, 3, and 4 different receptors on the same syncytium. So no, it's not an exception, but we have to talk about insects. For mammals, the story is different. And is there something special ethology about these pairs, like do the corresponding? So what is special and what has been shown that they are quite special is for example, for the pheromones. That in that case, they are quite special because they are coming usually together, and they can interact, for example, in giving the information about the female, the male, and where it is already a mated female. So this kind of information has been already shown to be important for different insects, moth, Drosophila. I don't know the details, sorry, so I will interrupt me. But these are the kind of information that you can see in literature. Martin, I hope that
So there's basis for it. All right. Thank you very much, Mario. Appreciate it.
Thanks to all of you. I will. Yep.
Sorry, I think I cut him off there. Clélia. All right. Our next speaker is Clélia Dumuletier, who will talk about population codes in the auditory pathway. And she is connecting. We'll be here. Clélia, I'll try you again. All right. There we go.
Okay. Yeah, that's really weird. I have a big message on the middle of my screen saying my connection has failed.
Huh.
You see me? You're seeing me, right?
I see you. That's right. Yeah. So a little bit fuzzy. And so I guess you can go ahead and share your screen.
I cannot actually because this message prevents me from doing anything. So I just see connection failed.
Oh.
And so maybe we can try again, disconnecting, reconnecting. I'm sorry.
Okay. Should I?
Yeah.
I'll close you and then reconnect you.
Okay. Thank you. Okay, right.
Hello.
Is it working now?
It's working. Great. Can you share your screen?
Okay,
All
can you
right.
see my slides?
Yes. We're all good.
Properly, or is it? Can you hear me properly?
Yep, we see it. Increase in the structure of the population code along the auditory cortical.
Great. So let me start. So I'm going to talk about the work I did at Penn with Vijay Balasubramanian and Yael Cohen. And the work was also all together with Lalita Suraya, who did the experimental work, Jane Lee, who did the identification of the areas and the sorting, and Jean-Hugues Lextang and Songhang Zhang, who did the analysis with me. So we are interested in the auditory cortex, and more particularly, we're interested in understanding if there are structural differences in the way information is encoded by the neurons in the different area of the auditory cortex. And so more particularly, we looked at two different big areas. So we looked at two core areas, which are the primary auditory cortex and the rostral area, and at two belt areas, which are the medium belt and the lateral belt. So they are here in red and green. And so the recording was performed by Lalita. She used a multi-channel electrode array. And the subject was a monkey, and the monkey was basically passively listening to different stimulus. And for this particular experiments, we presented two dynamic moving ripple to the monkey. So dynamic moving ripple is a very long sound, which is like about 5 minutes of stimulus. And it's composed of, it's like a very structured, kind of very structured noise that goes through all possible frequencies and all possible modulation of frequencies. So the monkey listened basically twice to that same stimulus. And so after sorting, we had about 20 recording in the core areas and about 18 recording in the belt areas. Each of them had between 8 to 21 neurons, which were single units. And so we asked different questions. And so the first question we asked was, can we analyze this data and study the functional connectivity between the neurons in each of these data sets? And in particular, we were interested in understanding how information is spread over the network of functional connection between the neurons. And by that, I mean, we are not exactly interested in understanding, we are not interested in exactly knowing which neuron interacts with which neuron, but we are more interested in general properties, such as is the information spread out over the entire network, or are there a few interactions that encode most of the information? Okay, so for that, we basically took the neuronal activity, we binned it in 10 millisecond timing, and we also used different size, time bin size. And in each bins, we had an activity variable, SI, which was 1 if the neuron was spiking and 0 otherwise. And we modeled this neuronal activity using maximum entropy model. So here I have represented the distribution of the maximum entropy model. So it's basically a probability distribution over the neuron activity. which has some parameters that are in green here, the G parameters, that can be either HI values or JIJs. And the first term in the exponential here, the HI times SI, they model an external drive for the neuron to spike or not spike. And the second term, which is a JIJSISJ, models a tendency of the neurons to spike coincidentally or at different moments. And so to do this modeling, we had to assume that the data was stationary. So obviously it's not true. But the idea here is to somehow find if there is some type of underlying structure of dependencies between the neurons. And finally, we are interested not in modeling all possible interaction between the neurons, but we are interested in finding if there is a minimal network of interaction that can capture all the pairwise pattern of the system. So how do we find a minimal network of interaction? So there are different approaches. The first approach we can use is called adaptive cluster expansion. It was developed by Simona Coco and Rémi Monasson. And so we used a code that is available on GitHub, on John Barton's GitHub. So the idea there is to expand the log likelihood function into the sum of different clusters of variables. And in this expansion, we will neglect the clusters with small contribution. And so that will allow us to remove a lot of interactions. And somehow that helps overfitting. The second approach we used is the approach I developed with Matteo Marsili and Paolo Mazza, which is the idea is to, it's some type of greedy method, which starts with no interaction at all. And we will add interaction one by one by selecting first the interaction that captures the most information from the data. And so we add the first interaction, we refit the data, and we look for the next most relevant interaction, and so on and so forth. So this type of greedy method is kind of feasible until about 20 to 25 variables. So, but the question in both methods is when to stop, right? So which cluster can we neglect and when to stop the procedure here? And so when to stop, we did it by balancing between goodness of fit and complexity. So in our approach, we consider complexity to be captured by how many interactions we have in the model. So the more interaction, the more complex was the model. And so the balance was made using this formula. So basically, we try to minimize the sum between minus the log likelihood and a complexity term that is proportional to the number of parameters in the model. So this approach is an information theoretic approach, and it is basically the first order, the first part of the minimum description length principle. And so we use this by doing this, we kind of prevent fitting noise in the data, and we really focus on what are the main interactions in the system. So here are some examples. In each of those boxes, so there are 4 boxes here. Each of them is a different data set. So if the first column was the fit performed on the first repetition of the DMR, and the second column is the fit on the second repetition of the DMR. And then the first line is performed with the ACE approach, and the second line with the greedy approach. So what you can see here is that networks are kind of similar between the ACE approach and the greedy approach, and they're also similar between the 2 repetitions of the DMRs. So if we want to have a bigger picture of what's happening here, I have represented here on the x-axis is the fraction of selected pairwise interaction in the first repetition of the DMR. On the y-axis is the same, but in the second repetition. And each dot here is a different data set. The green dots are data sets that are in the belt area, and the red dots are data sets that is in the core areas. So what we can see here is that, and the error bars are the differences between the 2 techniques. So what you can see here is that the network, generally the density that we find is consistent between the 2 techniques, and is also consistent between the 2 repetitions of the DMR. And finally, we can find that the networks tend to be quite sparse. So in general, for example, for 10 milliseconds, we have less than 40% of pairwise interaction that are selected. And this is true across time binning. So if I go from 1 to 50 millisecond timing, we still find very sparse networks. But how much of the information is extracted by these very sparse networks? So what I represented here on this graph, on the y-axis is the fraction of information that is extracted, is actually the fraction of pairwise information extracted. So I take all the information I can extract with a pairwise model, and I looked at how much information of that information is extracted by my 40%, let's say, interactions. And what we can find is that most of the pairwise interaction information is actually extracted by those less than 40% interactions. Actually, more than 80% of the information is extracted. And you can see this is true across time bin as well. And so basically we have very few interaction and encode most of the information that is available. And so as a result here, we see that we have a consistency between repetition of the stimulus. We observe that the networks tend to be sparse, and those sparse networks tend to extract most of the pairwise information that is available. And then if we compare between the two areas, the core and the belts, we find that belt area tend to be denser than the core area. So this somehow suggests that correlation patterns are more prominent in the belt area than in the core areas. And this is true across time binning as well. And so another question we can ask is, what about higher order pattern of the data? So right now I have discussed pairwise patterns, pairwise pattern of the data, but what about higher order patterns? So what we can see is that the fraction of the information the fraction of the total information available in the system that is captured by the pairwise model that we have considered is actually very small. So there is some higher order information here that we don't capture. That could be noisy information. It could be spread out over all possible higher order pattern or not, but this is something that still needs to be studied. So the second approach we had was to consider, was to try to study if there are a group of neurons that are highly correlated with each other. So for example, if I consider all my neurons in my data sets, we can ask if there are some groups of those neurons that tend to be more correlated with each other, not necessarily by pairs, but really by group. And so those neurons are not necessarily spatially close to each other. And so this is interesting in the case where we would like to apply some type of coarse graining and, for example, ask from higher order if we would like to study the system with kind of a higher order level. So for that, we use two different techniques. Also, the first technique is a statistical technique using dimensionality reduction. And the second technique is using still maximum entropy model, but this is a different type of maximum entropy model, which I call minimally complex model. And they are discussed, the technique is discussed in the paper that is shown here. So the first technique is a dimensionality reduction. So the idea here is that you will take your data sets, you will center it and you center your data sets, and then you will compute the eigenvalue in the eigenvector of the covariance matrix of your data. And then you will look at what are the most significant eigenvalues, and the number of most significant eigenvalues, they correspond to the number of assemblies for us. And then you can reduce your data set to only this significant subspace. And the second part is to apply an independent component analysis in order to, so you look in your subspace and you will reorientate your basis vector in order to go in the direction where your direction are not the most statistically independent direction. And this will define an assembly. And so this technique is described in the paper that is displayed here. The second technique is using maximum entropy model. So you see here the distribution of the state of the system that we consider. So it's similar to the one I introduced earlier with pairwise model. But now the phi mu here, they can be of pairwise, they can be S1, S2, they can also be of three-body, S2, S1, S2, S3, or even higher order pattern. It doesn't matter. You can consider any possible type of pattern. So what are minimally complex models? So minimally complex models are a subset of all those possible type of high order pattern models. And they look like that. They look like kind of communities. So you consider all your neurons and you will partition your neurons in different communities. And within each community, you put all the possible interaction. So now the idea is that you will find which type of this community model is the best fit in your data. And for that, we use Bayesian inference. So basically, we're trying to compute the models, what is the probabilities that each of these community model reproduce the data. And this is nice because it makes a balance between goodness of implicity. And in that case, this probability is actually very easy to compute. Okay, so there is advantages in both cases. So in the statistical approach, that is an approach that has already been used in the auditory cortex. And it's also nice because you can, in a new basis, you can rewrite a coarse-grained activity. So you can write an activity for your newly uncovered assemblies. On the other hand, for the modeling approach, the advantage is that you take into account how are the pattern of the data to compute the groups of neurons that are highly correlated. And the second thing is that it's a modeling approach. So you get a model at the end of the day. You get some type of coarse-grained representation of the dependencies in your data. And finally, these type of models are actually low complexity models. So they tend to be very simple model, and they will allow you to find a very stable pattern of your data. So these are some examples. On the left-hand side is using the dimensionality reduction. On the right-hand side is using minimally complex models. This is one dataset that has 10 neurons, and so what you can see is the first row on the left-hand side is after PCA, and the second row is after ICA. And so what you can see is that the assembly detected on the left-hand side are similar to the one detected on the right-hand side. So they have been inverted. Number 1 and 2 have been inverted. And so this is another representation. Here I also compared, this is another data set. Here I also compared with the pairwise analysis for that data set. So the first row is the first repetition of the MR, the second row is the second repetition. The left column is the pairwise analysis, the middle column is the dimensionality reduction analysis, where you find 2 assemblies, the orange and the blue assemblies here. And the right-hand side is the minimally complex model technique, and we also find 2 assemblies, and you can see that the orange and the blue assemblies are very similar to the one found with the dimensionality reduction. One thing that I would like to note is that the dimensionality reduction allow you to have the same neurons in 2 different assemblies. So for example, neuron 6 is in the assembly blue and in the orange assemblies. The modeling approach does not allow that. However, it allows for higher the pattern, and what you can see here is that the neuron 6 is put in the assembly in the blue communities, but is connected through an important 3-body interactions to neuron 8 and 10.
We have to get towards the end pretty quickly here. Thank you.
Yes. So to conclude, if we compare the 2 analyses, we found very similar features between the 2 approaches for communities, and we also find that assemblies tend to be sparser in the belt area than in the core area. So which means that neurons tend to be driven by fewer neurons in the belt than in the core. So as a conclusion, we propose different approaches. We propose to balance between fitting and complexity, which was not really, I think, not so much explored before. And we also propose a new approach to model coordinating neuronal activities. And comparing auditory cortex areas, we found that belt area tend to be denser than core areas, and we also find that they tend to be denser in terms of network, but sparser in terms of assemblies, which means that they are driven by fewer neurons than the core areas. So right now we are waiting for more data and to kind of explore a little bit more those different analyses. Thank you very much, and I'm back here.
Thank you, Clélia.
Sorry, yeah.
That's good. We have one question of clarification. Michelle Nardin is asking, when you talk about total information, do you mean the mutual information between stimulus and spiking, or are you working just off the spikes? And how do you estimate
So
that?
when I mean, exactly, so that's a good question. So when I mean total information here, I'm basically meaning what is the total, I'm comparing the total amount of information I can extract with a complete model. So basically, I'm comparing the Kullback library divergence between the data and a complete model versus the data and the model that I'm using. And I'm checking how far is the model I'm using from the data versus how far is a complete model from the data. So it's not the mutual information, but it's something that is more similar to a fraction of difference explained, for example. I don't know if that answered the question. Yeah.
Well, Michelle's asked a second question. She'd like to know, are minimally complex models based on correlations only or noise correlations? Do you take into account single cell preference or stimulus and afterwards interactions?
So the minimally complex model, they are based on all possible So they are based on everything that is in the data. So it includes also the noise. But the idea is that because we are doing this the comparisons. So we're doing we're doing Bayesian model selection. So the model that will be selected, the community that will be selected, will really balance between noise and the actual patterns of the data. So normally, the final model that we select should be able to discard whatever is noise in the data and only focus on important correlation in the data. I'm not sure, do you take into account single cell's preference for stimulus? I'm not sure I understand that part. I don't know if you want to specify, to be more specific in your question.
I think maybe she's asking whether you take into account single neuron response curves, tuning curves, for instance, at frequency.
No. No, we're just taking into account the spiking activity itself, and that's it.
Okay.
Yeah.
All right, I think we need to move on. Thank you so much, Clelia. It was a
much.
great talk. I really appreciated it.
Thanks.
And so our next talk is from William Barnett. Who will talk about the striatum and reward-based learning. Hello.
I'm trying to share my screen. Great.
Great. All right, I'll let you take it from here. Looks good.
Hello, my name is William Barnett. I'm a postdoc at IUPUI. My co-mentors are Alexei Kuznetsov and Christopher Lapish. Today, I'm going to talk about how striatal compartments participate in multimodal and concurrent reward-based learning. In the classical theory of basal ganglia learning, dopaminergic inputs from the substantia nigra to the striatum induce plasticity in D1 receptor and D2 receptor expressing medium spiny neurons to inform action selection. D1 receptor expressing MSNs experience long-term potentiation following dopamine release or following exposure to dopamine, and D2 receptor expressing MSNs experience long-term depression following exposure to dopamine. In this way, subsequent action selection attempts reinforce the circuitry that's associated with a rewarded behavior. We are interested in how some specific compartments of the striatum contribute to different aspects of behavior. There's been, over the last 20 or 30 years, some new research that has investigated what are the different roles of the medial and lateral compartments of the dorsal striatum. The dorsal medial striatum, or the DMS, is involved in goal-directed learning, and it's thought that the corticostriatal synaptic weights in the DMS embody reward outcome contingency. The dorsolateral striatum, or the DLS, is involved in habitual behavior, and it's thought that the corticostriatal synaptic weights within the DMS embody some learned stimulus response associations. The DLS is also a little bit slower, or it's involved in overtraining. Yeah, so this has kind of been a hot topic of research in the past couple of years. There are some machine learning style reinforcement learning models that get at how is habitual learning done and what is the DLS doing. There's also an interesting recent paper with Michael Frank about wave-like dopamine release in the dorsal striatum that gets into the different timing dopamine release in the dorsal medial and dorsal lateral striatum. These two compartments receive different inputs, both from the substantia nigra and from the cortex. This is a cartoon from a Dieseroth paper. I'm going to talk about this orange tan structure first. This is just a depiction of the kind of reward feedback that's experienced by the dorsal medial striatum. This is classical reward prediction error, where in response to a rewarding stimulus, there's an increase in dopamine release from the substantia nigra, and in response to an aversive stimulus like some kind of shock, there's a decrease in the amount of dopamine released from the substantia nigra. The DLS, however, in the DLS, dopamine release is encoding something else. You see that in response to an aversive stimulus, there's an increase in dopamine release. We're calling this, we're equating this with the expected reward, but people are using words like motivation and salience to describe this. It's indicating that this stimulus has been involved in learning. So what is my computational paradigm? We're doing brain simulations of 2 alternative forced choice tasks. This is a pretty classical way to represent the basal ganglia with concurrent loops through the cortex, the basal ganglia, and then back to the thalamocortical loops. I want to draw your attention to the purple structure first. This is the loop or the channel. I'm going to call it a channel that represents a specific action, like action, we'll call it number 1. This loop has its own direct pathway and its own indirect pathway. So it passes through, there are neurons within the direct pathway and indirect pathway medium spiny neurons that participate in this channel. And then following the output of the basal ganglia, it inhibits or disinhibits a thalamocortical unit that's specific to a particular action. By stacking multiple such loops, you can see I've added a blue one here, we can represent multiple actions. Since each channel has its own direct and indirect pathway, the representation for each activate can kind of turn itself on and off independently. We are going to extend this class of models to incorporate 2 cortical and striatal, or 2 cortical compartments and 2 compartments of the basal ganglia. The medial partition includes the prefrontal cortex because the dorsomedial striatum receives primarily input from the PFC, as well as the part of the basal ganglia that includes the dorsomedial striatum. The lateral partition includes what we're calling a premotor cortex, which is responsible for behavior, as well as the dorsolateral striatum. The dorsolateral striatum receives primarily cortical input from sensory and motor regions. This is the model is ordinary differential equations. Each one of the populations here is represented by a single ordinary differential equation that determines its firing rate. As discussed earlier, we've implemented the model to reflect reward prediction error in the DMS, and so this box just indicates that the corticostriatal synaptic update depends on reward prediction error and embodies reward outcome contingency. In the DLS, I've drawn a green box around it to show that this signal is the expected reward. We interpret it as salience, and it embodies stimulus response association within the DLS. Since I have 2 cortical compartments, this model is producing 2 choices. The choice that's made in the PFC we're calling the outcome, since it's informed by reward outcome contingency, and we're calling the choice that's made in the PMC the action, since it determines reward feedback. It defines behavior in this model. What is the MEL output? So this is an example of a pair of behavioral sessions. In the first block, I'm showing you initial learning. The firing rates here, that each data point here represents a firing rate of a PFC or PMC at the end of a simulation. And then by performing many trials, we update synaptic weights so that the model can learn something. So for example, at trial number, this is 150 trials before the change in the reward rule. You can see that the PFC firing rate for the blue population is high, so the PFC has selected outcome number 1, which corresponds to the color blue. And something similar happens in the PMC, and you can also see scenarios where the orange population in either the PFC or the PMC wins, and action or outcome number 2 is selected. I'm going to talk first about an initial learning block. Here, the agent is learning to select the rewarded action, which is action number 1, which corresponds to the color blue. I'm going to show you several of these performance plots. The performance is a number between 0 and 1, indicates how likely the agent is to select action number 1 or action number 2. So when this quantity is close to 1, the agent is primarily selecting action number 1. When it's close to 0, the agent is selecting action number 2, even if action number 1 is the incorrect action. So here, the agent learns pretty well to perform action number 1. The PFC is learning to do this a little faster at the beginning. Its uptake is a little quicker because it's using reward prediction error derived from goal-directed learning. The PMC needs to be taught at first, so it's a little bit slower. However, at steady state, the PMC has better performance than the PFC. We're going to quantify these two metrics later. Following an initial learning session, we do a series of follow-up sessions, including reward reversal, reward devaluation, and punishment learning. And so you see here, there's a change in the rule, and the agent has to learn to select the orange action. So what are these behavioral challenges? I don't have time today to talk about devaluation. Reward reversal is where we change the reward feedback so that action number 2 is rewarded instead of action number 1. In punishment learning, we change the feedback for action number 1 so that it has a negative valence. We're calling this negative reward a punishment. These are the results of reward reversal. So the agent already knows how to select action number 1. Now it must learn how to select action number 2. The medial partition, which uses reward prediction error, is pretty good at picking up that something has changed. It slips into an exploratory mode, and then consistently the PFC is selecting action number 2. However, the PMC needs to overcome its learned stimulus response association. The PFC has to teach the lateral partition how to do this. So we have to learn in the PFC and then teach the PMC, and it takes a lot of time. Again, similar to initial learning, at steady state, because there's a strong stimulus response association, the PMC is doing a better job than PFC. In punishment learning, there's some similarities and some differences from reward reversal. Again, the medial partition picks up pretty quick that something's going on because it's able to utilize a reward prediction error, and it consistently selects action number 2. Similar to reversal learning, the PMC has to be taught. It takes it more time to pick up on the change in rule. But note here that at steady state, the PMC performance is not as good as the PFC performance. This is because, in essence, the agent is learning to minimize the expected punishment. So the expected reward, the quantity there, is very close to 0. This is the quantity used for synaptic weight updates in the DLS, so there's no new stimulus response association formed. That's why the performance of the PMC is less than the performance of the PFC in this scenario. Our research groups are interested in substance abuse disorder, and impairment encoding in the PFC is thought to underlie a number of neuropsychiatric disorders. We're going to implement the loss of executive control and then repeat the punishment learning and reversal learning simulations. What does that mean? So basically, what other populations see as the output of the PFC, we do a weighted average of the signal that they're supposed to receive and then the signal that the opposing channel receives. And you can see the weighted sum. It's really simple here. But what it means is that learning in the DMS is not done with perfect information. It's muddled a little bit. Yes, so I just described this text, sorry. These are the change point analysis results of all these simulations. So we use a normative model of evidence accumulation for change point detection in the reversal and punishment sessions. This just tells us, like, when can we detect that the agent has learned to do a new rule or has learned to perform in the face of a new reward feedback rule. So I'm going to recapitulate here the difference in, like, how fast does it take the medial partition and the PFC and the lateral partition, which includes the PMC, to learn the new reward feedback rule. So we can compare these two black box and whisker plots as shown in the performance plots before. It takes the PMC several more trials to figure out that something different is going on. And this is recapitulated in the black box and whisker plots for punishment. The red box and whisker plots are following this corruption to the signal from the PFC, impaired PFC coding. There's a modest reduction in performance of goal-directed learning, which you can see here and here. The prefrontal cortices don't perform quite as well, but this translates to a much larger difference in the performance. The lateral partition, which you can see as a change point trial number for PMCs, there's a big difference between the two different simulations. So we interpret this to say that behavior is less flexible. This is the emergence of inflexible behavior. We're also looking at steady state performance. When I'm plotting, the black box and whisker plots indicate what is the performance at steady state of the prefrontal cortex, and what is this, and the gray box and whisker plots indicates the performance at steady state for the premotor cortex. In the simulations where a new stimulus response association is performed, so the DLS is able to learn very well, you can see that the PMC performance leads. So looking at this initial learning and the reward reversal simulations, the PMC performance leads. In simulations where there's no new stimulus response association learned, such as the punishment simulation, goal-directed learning is driving learning, and the lateral partition is kind of following, so PFC performance is greater. We're really interested in these results in the initial learning and reward reversal sessions, because since this goal-directed unit is no longer directly engaged in producing the behavior, the stimulus response association kind of takes over, the PFC is free to sample alternatives. So it will from time to time pick the wrong solution. And if the PMC picks the rewarded solution, there's no like big consequences for PFC for doing that. We interpret this. This could be a mechanism that drives something like tonic behavior. It could promote flexibility if the animal's trying to like monitor the reward feedback rule to see if it's changing. In the sets of box and whisker plots on the right, we're looking at our impaired PFC simulations, and in both cases, for example, in the reward reversal simulations, the difference between PFC and PMC performance is exacerbated, and in the impaired PFC simulations of punishment learning as well. So in conclusion, we have a new biophysical model of the basal ganglia that includes both the dorsomedial and dorsolateral striatum. To our knowledge, there's not a biophysical model that includes these compartments before this one. The dorsomedial striatum we show how it's critical for goal-directed behavior and sensitive to devaluation, and how the dorsolateral striatum is critical for stimulus response association and habit formation. Working together, these two compartments produce adaptive and flexible behavior. However, when we interrupt executive control by confusing the prefrontal cortex, we have experienced a decrease in efficacy of goal-directed learning in the dorsomedial striatum, and there's greater resistance to devaluation in the dorsolateral striatum, and this could be responsible for the emergence of inflexible behavior. I want to thank my co-mentors, Alexi and Chris. I want to acknowledge our funding sources from the NIH, and the Lilly Endowment has supported the supercomputing cluster that I use to do my simulations. Finally, I skipped a lot of technical details in this talk. If you're interested in sort of digging into it, you should check out our paper on bioRxiv, and this is a QR code that links to that.
Thank you so much for your talk. Lots to think about here. I mean, I guess we can read your bioRxiv. I think we have time for a quick question or two. So we had two questions about thalamus. One question about what part of thalamus is included here. And I thought I saw a second question about thalamus. We'll go with that one.
Yeah, so I see the question. We're not explicitly modeling the thalamus. This is, I mean, we acknowledge that these pathways go through the thalamus, but we're not like looking at any dynamics there. And so we, if you look at my diagram here, I have labeled this little shell, but I consider these like the units that produce behavior to be sort of corticothalamic units that are acting in synchrony. As far as continuous learning problem, yeah, so when I say that for example, in reward reversal learning, the lateral partition has to overcome a stimulus response association, it is forgetting that stimulus response association. That also depends on the time scale. On a shorter time scale, those synaptic weights could still be present and active.
Okay. So Siddhartha would like to know about the nucleus cumbens and ventral striatum involvement in habit formation and addiction.
So those things are involved, but we are looking at the dorsal striatum. There are some, okay, so let's go up to here. So I'm looking specifically at some publications where they are destroying or inactivating the medial or dorsal striatum. and showing how, for example, if you destroy the dorsolateral striatum or inactivate it, you can recover more quickly goal-directed learning, or vice versa. You can get stuck in habitual responding if you destroy the dorsal striatum. There's also some similar publications about addiction in that context, and that's what we're focusing on in this paper.
Okay, I hope that answers the question. Thank you so much for your talk.
Thank you.
And thank you. So next we have a talk from Scott Rich on neuronal heterogeneity and how that alters epilepsy. And I will invite Scott on screen. Oh, I have the wrong talk. Thank you. Scott is actually the featured talk at 11:40. Sorry, you're right. It should be Alessandro. My apologies, everyone. I'm looking forward to Scott's talk. So Alessandro will tell us about muscle activity during reaching. And here he is.
And I can't really hear you. I can see you, but I can't hear you.
I think we can hear you. So we'll do with that. So if I'm going to ask you to share your screen. Uh-oh. Alessandro, can you hear us now? Ah, there's your screen. Okay, can you try talking? Are you saying things?
Yeah. So can you see my screen?
I can see your screen and I can hear you, so I think we're all set to go. All right. And here's Alessandro.
So I can't really hear you, so you should maybe write in the chat if you can see my screen. That would be great. Great. Can you also hear me so that I can maybe start talking and then? Okay, so I'll just start talking, and then at the end we'll figure out how to deal with the Q&A because I can't hear the chair so far. All right, so hi everyone. My name is Alessandro Sabatello, and today I'd like to talk to you about my work on the composition of muscle activity during reaching movements into latent space, time, and action factors. So yeah, multi-articulated bodies such as ours, those of non-human primates and those of humanoid robots are very complex systems to control. In these systems, even simple motor actions such as reaching for a cup potentially requires the coordination of hundreds of degrees of freedom. For example, there are 240 in the human body to be precise. So yeah, and yet despite this complexity, evolution has converged towards a solution that in a way gives our motor systems the ability to deal very well with such complexity. As a matter of fact, biological motor systems are so capable that we are not only able to effortlessly reach for a cup, but we're also able to perform much more complex movements such as these. The way this is accomplished is, however, still a mystery to us. In fact, to the best of our knowledge, we still cannot really get to have a system that performs something similar to that. So our best engineering efforts, as a matter of fact, often lead to results such as these, that if we want to be generous, we might call suboptimal. And yeah, so what's also fascinating is that we are not only able to control such complex systems effortlessly, but we're also very good at generalizing. For example, once we have learned how to walk on a flat surface, we can also easily walk on the sand or on the snow. And similarly, if we know how to control our body to reach for a cup that is placed on the left of a table, we most likely also have no issues in controlling our body to reach for the cup if this is somewhere else. For example, if it is right in front of us or to our right. So it almost looks as if our motor system can quickly, adjust some parameters and perform new actions it never performed before. So how does the motor system accomplish that? So we can identify. Can we identify the key computational principles that it uses to deal with the inherent complexity of controlling multi-articulated bodies? A classical approach that has been to address this question is to record and analyze the output of the motor system, that is, the spatiotemporal patterns of muscle activity during the execution of some movements. These analyses have revealed that generally the motor system does not control muscles individually, but it controls them in group. The first direct evidence comes from Thresh and others, who show that if you take a spinalized frog and you stimulate its limb in 14 different locations, you'll observe very structured muscle responses. For example, the muscles ST and SA are often active together, regardless of the stimulation site. And the same is true also for other muscle groups, such as AM and VI, but also BF and IP. And subsequent experiments even prove that, of course, similar regularities are observed across a wide range of behaviors and animal models. Importantly, subsequent work by Levine and others later provided convincing evidence that some premotor neurons in the medial deep dorsal horn appear to support such organization. So this, in a way, tells us that the statistical regularities observed at the level of muscles, in a way, reflect some concrete fixed structure in the spinal cord. So how do we model such regularities? Well, basically, the most popular way in which such regularities across the muscle space have been captured is through very simple linear models. These models assume that muscle activity Y, recorded on muscle M during action theta at time t, is simply the sum of the contributions of a fixed number of spatial modules AI, each activated by an action-specific motor command UI. So this model, as you can imagine, is basically equivalent to a simple feed-forward linear network, where the spatial module is defined by an input neuron and together with its output weights. So now, this model is, of course, suggesting an effective way to capture regularities in the spatial domain. However, at the same time, it introduces significant complexity in the temporal domain. In fact, this model assumes that for each new action theta, represented by different color saturation here, the motor system has to specify a completely new, basically, motor command, which might not be an optimal strategy. So we can say that this model, in a way, is space-centric and introduces temporal complexity. Other studies have focused their attention on the temporal domain and have also observed significant regularities. So, for example, Ivanenko and others recorded the muscle activity of 16 leg and trunk muscles of 6 participants as they walked on a treadmill at different speeds, and they observed that basically 5 temporal modules could account for large fractions of the total variability in the original dataset. And also in this case, subsequent studies, electrophysiological studies, in this case on the spinalized frog, this time conducted by Hart and collaborators, provided evidence that suggested that the temporal regularities are also mediated by specific circuits in the spinal cord. So also in this case, the way such regularities are captured is through simple linear models that assume, in this case, that muscle activity Y is the sum of activities of the fixed number of temporal modules UI, whose contributions are weighted this time differently depending on the action theta. So this model, as you may have guessed, can also be interpreted as a simple 2-layer feed-forward neural network. Now, this time, the temporal modules are simply some fixed inputs to the network. What's interesting is that now, while the previous model was only able to capture spatial regularities, this model is only able to capture temporal regularities. In fact, this model is introducing significant complexity in the spatial domain, and in fact, it assumes that for each new action, the motor system needs to specify a completely new matrix of spatial weights to map the motor commands to the muscle activity. So we can say that this model, this kind of model, is time-centric, and it introduces spatial complexity. So can we do better? How do we capture both spatial and temporal regularities? So recently, Dellis and others proposed a model that is able to do so. This model interprets the muscle activity Y as being generated by fixed spatial and temporal modules, A and U, that are flexibly combined depending on the action. And this is done through some action coefficients B. So now the space by time decomposition model, which is what this model is called, can also be interpreted as a linear free-form network. But now the network has 3 layers. So now this model is able to capture both spatial and temporal regularities. However, it makes the strong assumption that the motor system specifies a completely new mapping between motor commands and spatial modules for each new action theta. So also this model, in a way, doesn't really propose a mechanism to simplify the control problem, and it introduces what we might call network complexity. So can we do better than this? So can we capture both spatial and temporal structure with a simple, biologically plausible mechanism that would actually simplify the control problem? So it turns out that we can. So I was thinking about this, and I thought that if we make the simplifying assumption that each temporal module is associated with a single spatial module, then we can decompose the muscle activity into the sum of contributions, this time given by the product of 3 terms, A, B, and U, but this time these terms only depend on action, on muscle, action, and time, respectively. Also, in this case, we have a simple interpretation. Actually, it's very interesting because now the network is very simple. So it is a 2-layer gain-modulated linear free-form network, and this time the gains are this time the action coefficients basically define the gains of this network. So now, according to this model, the motor system can generate a completely new action by only modifying the gains of the input neurons. So it does not need to specify new motor commands or completely new mappings between input neurons and muscles, or between temporal and spatial modules, such as was proposed by the previous models that I've showed. So, well, yeah, what's also more very interesting is that now the corresponding network So what's more interesting, as I was saying, is that the network is proposing a mechanism that is based on gain modulation. And gain modulation is not a rare phenomenon, but is considered as one of the main computational principles of the nervous system, and has been object of many influential reviews and seems to play a major role in many processes requiring transformation of coordinates, which is promising. But now we only have a model. So actually, it's a very simple one. The question now is, how good is it? So to validate the model, I used a recently published dataset of muscle activity data recorded from healthy participants during the execution of reaching movements in the frontal plane. Specifically, the participants were asked to reach to 9 different targets placed in front of them. And this basically gives us the possibility to look at the muscle activity generated during the execution of 9 different actions. So after preprocessing the data, the first thing that I did was to fit the gain modulation model and also the other less constrained models to the individual subject data. So as a reminder, the gain and modulation model is the most constrained and less flexible one of those that I've talked to you about today. So it is not guaranteed that it will be able to explain the data well. And yet it turns out that it does. So here I'm showing the ground truth and the reconstructed muscle activity of 3 representative targets and muscles. And you can clearly see that there is a fairly good match between ground truth and predicted data. And also, of course, on average, the model fits the data as well as the classical modular models, despite its lower number of degree of freedom, which is also good, of course. So now that we know that the model fits the data well, we can start looking at the representation learned by the model. So for example, we can start looking at the temporal and spatial modules learned by this network, which are plotted here. And we can ask whether these modules are in a way similar to those learned by the other models, which have been intensively characterized over years and years of investigations. And yes, it turns out that they generally are. So in fact, if we ever look at the representation learned by other models, such as the space-centric one, or also the time-centric, or the space-by-time one, you can immediately see that, for example, the learned spatial modules are quite similar across models. And the same is true also for the temporal ones. So next, I looked at the representation of the action manifold learned by the model. And this is important because, if the model, if the motor system uses spatial and temporal modules to simplify the control of movement, then it needs to be able to recruit them flexibly so as to perform a variety of new actions. So let's have a look at it. So I will show you the action factors B, that is the gains of the input neurons, as a function of the reach directions. And here they are. So basically what these panels show is that the first two factors are approximately always recruited. So we can say that they are weakly tuned for reach direction. While on the other hand, the other three factors are very strongly tuned. Specifically, as you can see, factor 3 seems to be mostly activated to reach to the top right target, factor 4 to reach to the bottom right, and finally the factor 5 to reach to the top left target. And so as you can see, these surfaces are very smooth. This means that they can easily fit by simple linear models, such as radial basis function models, and which is something that I did. And here I'm showing you the predictions that, as you can see, also fit the data quite well. Moreover, it turns out that the action representation manifold is so smooth that we don't really need to observe all the muscle patterns to learn it. So to prove this, I've performed a leave one out cross-validation. That is, I've learned both the gain modulation model and the RBF model only using 8 out of the 9 actions. And I've tried to predict the muscle patterns for the action that was not used during training. And of course, I've repeated this procedure 9 times, each time leaving out a different test action. And as you can see, this procedure works quite well. So here again, I'm plotting ground truth and prediction for 3 representative muscles and 3 representative targets. And you can see that there is a quite well match. So in a way, this means that we don't really need to explicitly learn to represent all the possible reach directions. Once we have learned a few, we can easily generalize and we can generalize, and with this we can generate new muscle patterns for untrained reach directions. So in summary, today I have proposed a gain modulated network model for spatiotemporal muscle coordination. I have also showed that the model decomposes muscle activity into spatial, temporal, and actual modules that are similar to those identified with traditional modularity models. And lastly, I've also showed that the model learns a smooth representation of actions that allow the generation of muscle activity patterns for untrained reaching directions. So finally, I'd like to thank my supervisor, Professor Martin Giese, and our funding sources. Thank you for listening. So now, not sure how to proceed because I still can't hear the share.
You can't hear me? No.
So maybe I can.
So I think if people can ask questions in there. Yeah.
To answer, you're nodding. No, what else I can do? Video.
Uh-oh.
I heard some typing. Okay, I'm reading a question. So, does your model work for more complex motions? Does it fail for some motions? So, yeah, this is a very interesting question. And the short answer is that we don't know yet because we haven't looked at it. But that's, of course, the next thing that we will do. So we will try to basically fit the same model to more complex motions. For example, we were looking at coordinated basically arm and hand movements, and we would like to see, if our model is powerful enough to capture the variability in muscle activity patterns there. So does it fail for some motions? We don't know yet. We have to figure it out. Concerning the 9 bridging directions that I've tested, I didn't see any significant variability across actions. So for the actions that we have looked at, it seems to be working well. What is new here? Well, okay, so gain modulation has been proposed. So the second question, gain modulation has been proposed a long time ago for the visual system. What is new here? Well, that's a good point. So what is good here is that what is new here is that I basically provide a unifying framework for very common theories in the motor control community. So there are these theories that date back 20 years or so, and each of them are based on, as I showed you earlier, on very simple linear models. And those models don't really talk to each other. They're only able to identify very specific mechanisms. So one class of models was only able to identify temporal regularities. Another class of models was only able to identify spatial regularities. Here, what I'm showing is that if we put everything into the framework of gain modulation, we can explain both sort of regularities. And then, yeah, so it just provides a unifying framework for long-standing computational theories in the motor control community. I don't know if that, in a way, addressed the question. Okay, well, thank you so much.
Starts with Scott Rich's advertised talk at 11:40. All right, so thank you for tuning. I shouldn't be muted. Okay, all right. Don't know what's going on here. All right. All right, thanks everyone, and we'll see you on our next session at 11:40.